Hello guys, let me introduce myself. My name is IJTIMOIY and I'm teacher of biological chemistry. Today we'll talk about metabolism and function of carbohydrates. Carbohydrates for heterogeneous organisms are the primary sources of energy. Digestible carbohydrates for the body include starch, glycogen, and disaccharides, is sucrose, maltose, and lactose. Monosaccharides are less common in the human diet. The function of carbohydrates. Carbohydrates serve as source of energy. Due to their oxidation, 55% of the entire human need for energy is satisfied. Here the main role belongs to glucose and glycogen. Carbohydrates are part of the structural and functional components of cell. Compounds of other classes, in particular lipids and some amino acids, can be synthesized from carbohydrates in the body. The most common carbohydrate in animals is glucose. It played the role of a link between the energy and plastic functions of carbohydrates, since all other monosaccharides can be formed from glucose and vice versa. Different monosaccharides can be converted into glucose. The classification and physicochemical properties of carbohydrates are studied independently. Carbohydrate digestion. Food carbohydrates in the digestive tract break down into monomers under the action of glycosidase enzymes that catalyze the hydrolysis of glycosidic bonds. The main sign of starch digestion is the small intestine, where the amylase enters as part of the pancreatic juice, resulting in the formation of oligosaccharides and the disaccharide maltose. Disaccharides, sucrose, and lactose enter the body with food. Oligosaccharides and disaccharides are hydrolyzed by specific glycosidases of the small intestine. The products of the complete digestion of carbohydrates, the monosaccharides, glucose, galactose, and fructose, into the bloodstream through the intestinal cells. The passage of glucose from the blood into the cell depends on insulin. This hormone makes the plasma membranes of cell permeable to glucose. The exception is the cells of the brain and liver. Phosphorylation of monosaccharide. The first chemical transformation of glucose in cell is its phosphorylation as a result of interaction with ATP. This reaction is catalyzed by the enzymes hexokinase and glucokinase in the liver. In other organs, only hexokinase. The reverse transformation of glucose 6-phosphate into glucose is also possible under the action of the enzyme glucose 6-phosphatase. Glucose 6-phosphate plus H2O, and we can form glucose and phosphoric acid. Galactose and fructose coming from the intestinal tract with the participation of the enzymes galactokinase and fructokinase, respectively, are phosphorylated at the first carbon atom. There are three parts to this process: glucose-specific transformations, glutaminating in the formation of pyruvate, aerobic glycolysis, common pathway of catabolism, mitochondrial electron transport chain. As a result of this process, glucose decomposes to dioxide carbon and molecule water, and the released energy is used for the synthesis of ATP. We have covered the second and third parts, and here we will consider the transformation specific to glucose. Aerobic glycolysis. The breakdown of glucose to pyruvate in turn can be divided into two stages: from glucose to glycerol dihydrogen phosphate, from glycerol aldehyde phosphate to pyruvate. Since the oxyacetophosphate is converted to glyceroldehyde phosphate. Reaction 5. Two molecules of glyceroldehyde phosphate are ultimately formed. The second stage of glucose breakdown includes reaction associated with the synthesis of ATP. Anaerobic glycolysis. In animal and human cell, the enzyme lactate dehydrogenase is widespread, catalyzing the reversible conversion of PVC into lactic acid. The cytosolic enzymes that convert glucose to pyruvate, together with lactate dehydrogenase, are able to provide ATP synthesis in the absence of oxygen under anaerobic conditions. In this case, PVC serves as a hydrogen acceptor which is converted into lactic acid. In the reaction, 4 moles of ATP are formed per 1 mole of glucose. A similar process in bacteria is called lactic acid fermentation. In this case, PVC is the first decarboxylated to form acetaldehyde, which is then reduced to ethyl alcohol. Regulation of glycolysis. The rate determining glycolysis in the enzyme phosphofructokinase, third reaction; hexokinase, first reaction; LDH, 11th reaction. Glucose biosynthesis. The synthesis of D-glucose from non-carbohydrate precursors is called gluconeogenesis. The synthesis of glucose from pyruvate occurs mainly in the liver, in the cortical substance of the kidneys, and in the epithelium of the small intestine. Gluconeogenesis basically follows the same pathway as glycolysis but in reverse. However, 3 glycolysis reactions are irreversible, and at this stage, the gluconeogenesis reaction differs from the glycolysis reaction of stage 1, 3, 10 of the reaction. The conversion of pyruvate to phosphoenol pyruvate, reversible stage, is carried out with the participation of 2 enzymes, pyruvate carboxylase and phosphoenol pyruvate carboxygenase. 2 other irreversible steps are catalyzed by fructose bisphosphate phosphatase and glucose 6-phosphate phosphatase fructose 1,6 bisphosphate. The overall process of gluconeogenesis is described as follows. Part of the PVC in the muscles is converted into alanine by transamination, which is transported to the liver and here again forms pyruvate, the glucose-alanine cycle. Regulation of gluconeogenesis. The central enzyme regulated gluconeogenesis is pyruvate carboxylase. Its positive allostatic modulator is acetyl-CoA. At the same time, it is considered a negative modulator for the enzymes of the pyruvate dehydrogenase complex. The second enzyme regulating gluconeogenesis is fructose 1,6 biphosphatase. This enzyme is inhibited by MP, however MP is a positive allostatic modulator of phosphofructokinase. Regulated by hormones, insulin inhibits gluconeogenesis, glucocorticoids, catecholamines, glucagon, pituitary hormones enhance gluconeogenesis. Fructose and galactose metabolism. Fructose is included in the glucose breakdown pathway at the 3-phosphate stage. Congenital fructose intolerance is associated with hereditary deficiency of fructose 1-phosphate aldolase. Influence of ethyl alcohol on carbohydrates metabolism. The metabolism of ethyl alcohol includes the hydrogenation reaction, as a result of which acetic acid is formed, part of which is converted to acetyl coenzyme A. In the liver,